connect properly for them. So yeah, I've never been down there. So yeah.
Well, good evening, everyone. Thank you very much for coming tonight. My name's Douglas Acodney. I'm a neurologist and neuroscientist here and director of the Neuroscience Mental Health Institute. I'm glad to be part of this interesting partnership night with the Alzheimer Society of Alberta Northwest Territories. And I think we're just about ready to begin. I want to thank everybody for coming, and I would initially like to acknowledge that the University of Alberta, its buildings, labs, and research stations are primarily located in the traditional territory of Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Shinnecock nations that are now known as part of Treaties 6, 7, and 8, and homeland of the Métis. The University of Alberta respects the sovereignty, lands, history, languages, knowledge systems, and cultures of First Nations, Métis, and Inuit nations. Thank you. Just a little bit about the Neuroscience Mental Health Institute. This is a little write-up that we did a few years ago. It's really a grassroots institute of the University of Alberta. It depends on a university and Faculty of Medicine and Dentistry, Faculty of Science for support. But it's additional work done by faculty on top of what they normally do during their day in their research and in their teaching. It's dedicated twofold towards understanding the complexity and beauties of the nervous system, which we'd like to convince you about, but particularly to prevent, attenuate, and eliminate suffering and disability. And I think this Alzheimer's Night is a really potent example of that. We work on brain, spinal cord, and nerves. The Institute is predicated on transparency, responsiveness, lots of internal dialogue and collaboration without boundaries in kind of a unique experiment. And we're a little different as an institute, especially with a major training component. I'll just quickly show you that there's multiple pieces to an institute. Alzheimer's and dementia research is a really important piece of it. And a lot of that resides in this Center for Prions and Protein Folding Disorders, two speakers of which are going to talk tonight with really cutting-edge research. Because what happens to proteins affects what happens to cells affects what happens to the brain, and it's a really key part of the story. But there's also other parts, and some of you may have had the opportunity to join us for other presentations. Sensory Motor Rehab Group, the MS Center, the Neurochemical Research Unit. SINAD was a collaboration with the Alzheimer Society that helped sponsor tonight. And Hope for Tomorrow previous grant, Dr. Dixon was part of that. The SMART Network and imaging. So just a little bit about what NMHI does. And with that, I think we want to show you a brief video here. No? Back. And then that one. Okay. Bear with me. Only neuroscientists.
The brain and spinal cord are the most complex and important parts of the human body. They control everything we do and drive us to feel, think, move, and learn. Hundreds of billions of interconnected cells in our brain and nerves, all acting in concert, allow us to experience the world and make us who we are. But there is still so much left to understand about this complicated and fragile organ. 1 in 3 Canadians are impacted by a neurological or mental health condition at some point in their lives. From Alzheimer's to multiple sclerosis to depression, these diseases and disorders are leaving a devastating impact on the lives of millions. Fortunately, there is hope in knowledge. By furthering what we know, we can find a way to diagnose earlier, treat more effectively, and implement preventative measures, easing the burden for those afflicted and improving their quality of life. Through the efforts of dedicated scientists and clinicians, we can work towards better brain health for tomorrow. At the Neuroscience and Mental Health Institute, we are dedicated to improving brain health through research. Located at the University of Alberta, we are home to over 150 world-class scientists and clinicians, all dedicated to the same goal: to better understand the brain and nervous system in order to develop transformative improvements for those affected by neurological and mental health-related diseases and disorders. By working collaboratively and taking a multidisciplinary approach, we are able to reach new insights and innovations faster. Through transparency and knowledge sharing, we are working together to make a difference.
I next have the tremendous pleasure of introducing one of the key persons that has helped identify the importance of an event like this and to make it happen, Dr. George Andrews. Dr. Andrews is CEO of the Alzheimer Society of Alberta and the Northwest Territories, and I'll just read you a little bit about, you're going to hear more from him, but the Alzheimer Society strives to improve the quality of life for those facing Alzheimer's disease and other dementias. In partnership with the Alzheimer Society Federation, the Society funds critical research, provides education and resources, raises awareness, advocates in partnership with government, private and nonprofit organizations for the treatment and ultimately a cure for Alzheimer's disease. Prior to joining the Alzheimer Society of Alberta Northwest Territory, Dr. Andrews was president and CEO of Alberta Cancer Foundation and vice president of external relations and chief development officer of the Northern Alberta Institute of Technology. He was a faculty member teaching leadership development to college and polytechnic leaders in Canada and abroad. We're glad we've moved him over to the Alzheimer field. I think we're very fortunate to have him working in this sphere. He holds a BA from the University of Alberta, an MA from Royal Roads University, a Doctor of Business Administration from the University of Bath, and is currently holding an adjunct appointment with Cape Breton University and has taught leadership and organizational change within the MBA program. So George, welcome. Thank you for helping with this event tonight, and I'll turn it over to you.
Thank you. I always find it slightly intimidating when people applaud before I say anything. So I appreciate the vote of confidence. I want to thank you all for coming here today, and I want to acknowledge the approximately 500 or so people that we have from across Canada and actually the United States and around the world. So it's quite an impressive outing for us. And so we've heard a little bit about why we're here, but I just want to hit on a couple of points that are important to me personally and to our society. In Alberta alone, 1 person in 3 will be directly or indirectly impacted by this disease. There are currently approximately 59,000 people living in Alberta with a diagnosis of dementia. I personally think this number is understated, but that's the facts we work on. A study was published last year by our national body that said in Alberta, in the next 25 years, the number of people living with dementia will increase by 300%. In 2019, a study indicated that 40 million unpaid hours of care and support were provided by informal care partners supporting people living with dementia. That typically would be another family member. That's the equivalent of 23,000 healthcare aides. Our society is really targeting on 3 broad outcome areas. Firstly, we work directly with people and their families living with dementia. We provide support and guidance to help them navigate on their own individual journeys. Secondly, we are focused on bringing a higher level of awareness and accommodation for people living with dementia in our communities, and we are actively encouraging increased funding for research. We're very privileged to have a university of the stature of the University of Alberta in our province and in our community, and they are doing research well above their weight in the international market. We have plans and we want to focus on having impact. A couple of examples are, recently we announced a $5 million fund to fund research at Alberta-based institutions. Our expectation is that this $5 million fund will actually be leveraged to $10 million over the next few years. We launched a Dementia in the Workplace resource to share information and provide guidance for employers to help people stay in the workforce longer, and also to help employees understand their rights and how they can stay engaged, because socialization and having a sense of purpose is very important for people living with dementia. We want to stay completely focused on and advocating for more research funding. By way of example, a 2018 report indicated that dementia research received the lowest research funding per capita in Canada at $5 per capita. By comparison, heart and stroke disease received $12 per capita, multiple sclerosis received $194 per capita, and ALS received $700 per capita in federal funding. Not to say any of these causes aren't important, but I think we need to put a voice to this. So I invite all of you, we're in Alberta, and perhaps beyond, people online, we know there's probably going to be an election coming up in the next few months. I invite you all to ask questions at your door about how important it is for this government to support people living with dementia and their families. If we can help, we want to help. So feel free to reach out and connect with us. Our services are free. You can access our website for personal services or for making connections. If you're out of province, please feel free to connect with your local chapter of the Alzheimer's service. Even if you're in the United States, there are chapters in almost every state. As I mentioned, we have an initiative to fund $5 million of research in Alberta-based institutions. If you want to be involved in helping to support that, all of our activities and actions are completely funded by donors. We're very privileged to have a very strong donor base, and for the people in this room who are donors, I want to say thank you very much. Without you, we wouldn't be able to be here. And lastly, I want to thank Dr. Zacconi for agreeing to host us and for inviting us to be here. It's been a real privilege for me, and myself and some of my staff will be around if you have any questions, and if you wanted to get involved in financial contributions towards research, feel free to reach out to myself, and Holly is over there, or reach out to us on our website. Thank you.
Great, thank you very much, George. We're going to have some time for questions at the end, so we have a Q&A at the end, so you can save your questions then for George and the other speakers. The Alzheimer Society also has a table out in the atrium with client service representatives who can help you after the lecture. We initially were hoping Dr. Roger Dixon could present at this juncture to give a kind of nice balance to the evening, because Roger is heavily involved in the Canadian Consortium of studies on Alzheimer's disease across Canada, sort of a clinical look at aging and what's happening to the population. But I think probably in the future we'll have a chance to have you hear Roger at a later date. This is my filler slide here, just before we introduce Dr. Sim. And this is hot off the press. I love this slide. This is a neuron, and it's sitting in a dish. But all these little spikes are specific molecules that are signaling this neuron to grow. And this is what we need. We need the connections to regrow, to reform, to prevent the cognitive decline in dementias or the other deficits and other neurological disease. So I couldn't help but showing that one slide. Next, I have the privilege to introduce my colleague in the Division of Neurology, Dr. Valerie Sim. Dr. Sim is an associate professor, Department of Medicine, Division of Neurology, and a member of the Neuroscience and Mental Health Institute. She self-identifies as a scientist who practices medicine to support her music habit. She's a very accomplished musician, and I told her I would tell the group about a performance coming up on the 12th, where you can go hear more of Valerie in her other role. But by day, she's a prion scientist. And again, prion is a key part of the protein abnormalities that we're trying to sort through what's happening in dementia, why these neurons are not growing and failing. And she's going to tell you about prions. And there's a building that's called the Center for Prions and Protein Folding Disorders, where key Alzheimer's and prion research is done. And there's overlaps with other kinds of disorders, and you'll hear names like chronic wasting disease, mad cow disease, and others, but very close relationships to dementia. She's a clinical neurologist and works in a cognitive clinic at the University of Alberta in the K. Edmonton Clinic. And by night, she's a violinist and fiddler. She's also passionate about promoting science communication and has published a very nice TED Talk on prion disease. And I think you can still get it up on YouTube. It's terrific. She regularly presents the science of prion disease to diverse communities across Alberta, Canada, and internationally. Her talk tonight will be about prions and Alzheimer's disease. And just briefly, so prions are these proteins that are folded the wrong way. And they're misfolded throughout the brain, ultimately lead to the death of brain cells. And the classic ones have a lot of strange names, like bovine spongiform encephalopathy, which is mad cow disease, chronic wasting disease, which is decimating deer populations. You may have heard Dr. Wille talking on CTV the other night about that research, but closely related. Creutzfeldt-Jakob disease, which is a condition that Val and I hate to see in hospital, but occasionally we do. A condition we have no cure for that causes a very rapid dementia, uncommon, fortunately. So prion diseases share many similarities with other dementias, like Alzheimer's disease. Dr. Sim's lab is interested in how the size and shape of different protein strains leads to the development of disease in different kinds of brain areas and different symptoms. A range of symptoms can also be seen in Alzheimer's disease and other dementias. And there's growing evidence that strains of Alzheimer's disease exist, also influenced by the size and shape of this one protein can cause such protein effects. It's an incredible story. It has implications for diagnosis and treatment. And I will stop and let Dr. Sim speak now. Thank you very much.
Great, thanks very much. I don't need to talk now. You said it all. You said it all. The one thing, though, we say prion. That's how you can tell a true prion researcher. Anyways, it is a great honor to be here tonight to speak to you. I really feel humbled to speak to people who come to learn more about this devastating disease. And I hope I can share with you the insights we're trying to gain through research and maybe inspire you to raise some of your own questions and dollars, I guess. So, prions and Alzheimer's disease, why size and shape matter. So let's start with what is a prion? Well, the easiest way to think of a prion is as an infectious protein. Now, what is that? Most of you are familiar with viruses, especially these days, bacteria. We know that they infect cells, they inject their DNA or their RNA, which is their genetic code, into the cell, make more of it, then wrap themselves up in protein and move on to infect other cells. But this case is a protein only that infects a cell, replicates more of itself, and moves on. So how is that possible? There's no genetic material. Good question. Well, I couldn't resist the opportunity to show my cute dog, Coda. When thinking about how to understand how a protein can spread, I wanted you to think about snow crystals, ice crystals, which we know well here in Edmonton, and seem to accumulate on Coda's face. If you think about an ice crystal, it starts very small, but then it attracts other crystals, and these crystals grow and spread, just like you'd see on your windshield. Well, this idea of spreading is very relevant to prions and proteins. So let me take you into the prion life cycle. So here's a cell, and on the surface of the cell is a protein. So instead of a water molecule or ice crystal, we have a protein. Now, this isn't the molecule that you add to your milkshakes to make them thicker. This is protein is something our bodies make and use all the time. And in this case, it's sitting on the top of the cell, and it has a very specific shape. It's a little folded into these helixes. In prion disease, something happens, and it changes shape. It misfolds, and it doesn't stop there. It sidles up to its neighbor protein, causes it to misfold. And this process continues, builds up in the cell, spreads to the rest of the brain, and winds up giving pathology and damaging the brain. What does it do? It causes holes in the brain. Now, in prion disease, these holes, as you can see in these little circles, these holes in the brain make the brain look a bit like a sponge. And that's why we call it spongiform change. And the name of this disease, prion disease, is often known as transmissible spongiform encephalopathy, which is a bit of a mouthful, but that's the reason, because the brain turns to a sponge. Now, these are some of the transmissible spongiform encephalopathies. The oldest one we know about is scrapie, which affects sheep. There are a number that affect humans. Kuru, Creutzfeldt-Jakob disease, as Dr. Zakhodny mentioned. There's some genetic forms. Of course, most of you will have heard of BSE, mad cow disease, and many of you are probably hearing about chronic wasting disease, which is on the rise and affects cervids. So you may be saying, fine, what does this have to do with Alzheimer's disease? Well, let me now introduce you to the prion family, because there are a lot of diseases that are very prion-like in this spreading misfolding. So if the prion protein misfolds and builds up in the brain, as shown here, you get prion disease. If alpha-synuclein misfolds and builds up in the brain, which is this purple blob, you get Parkinson's disease. And guess what? Amyloid beta and tau, two proteins, misfold and build up in your brain, you get Alzheimer's disease. So what can prion disease research tell us about Alzheimer's disease? Well, what I'd like you to come away with today is an understanding of strains, the concept of strains. Most of you have heard of that with COVID and all the various strains. Here, it's a little bit different. It's just a protein. How can a protein have different strains? Well, if we go to a simple imagination of this protein, little curlicue, if I were to misfold it or change its shape, I could actually fold it into a number of different shapes. Say you have a paper clip and you twist it and refold it. And interestingly, different shapes of this same protein will cause different patterns of disease. This is what we talk about when we say a different strain. It causes a different symptom, a different pattern. So in the research world, if we're interested in shape, we have to say, well, how can we see shape? These are pretty small. Well, we need technology, and one of the cutting edge technologies we're using now is cryo-electron microscopy. And my colleague, Dr. Wille, is an expert in this area and is on the leading edge of many of these discoveries. What is the shape of these proteins that are misfolded? Well, here's a couple of prions for you. These are the actual cryo-EM micrographs of prions. And if you imagine it's like a paper clip that's kind of twisted, and I don't have to work on my prion, shape thing, but anyway, there are different shapes for the different strains of prion. Same protein, different shape, causes a different looking disease. What about Alzheimer's disease? Guess what? Same technology. This is amyloid beta from Alzheimer's disease. And there are different patterns of it as well. It's a bit more like this sort of thing happening. Tau, the other protein in Alzheimer's, it too folds into different shapes. So shape is important. It affects the type of disease you get, whether it's prion disease or Alzheimer's disease. What about size? Well, let's talk about size. So imagine I have this misfolded paper clip. Well, remember the seeding, the growing, the templating, spreading? Well, it can form a bunch of different sizes of particles. And guess what? Different sizes of particles also influence what the disease looks like. Okay, so coming to the research, how do we measure size? Pretty small things. Well, here we use something that's gently known as asymmetric field flow fractionation with inline scattering. Or we just call it AF4. And essentially, this is about fluid flow. So bear with me. I'm going to take you through a little physics. This is a tube, empty tube, like you would water the lawn. I'm going to flow water down this tube. With me so far? Well, when you flow water in a tube, the flow in the middle of the tube is faster than the flow on the side. Think of the river. If you go to the river and you watch it flow by, it's the middle of the river that's really flowing quite quickly. Well, we take advantage of that in this system. So we flow some fluid down one direction and at the same time the other direction to sort of trap the flow in the middle, and we add particles of different size. In this case, it's actually brain material. And we've done this with patients who've died from prion disease and from Alzheimer's disease. We put the brain material of different sized particles in here. We pass yet another flow across these particles, and it pushes the big ones to the bottom. Think of them at the bottom of the river, and the smaller ones stay in the middle. So what we've done when we stop one of the cross flows, we let the particles flow down the river. The little particles are in the middle, so they come out first, and the big ones come out later. Then, once we separated them by size, we pass them through various laser lights that bounce off the particles and give us information about their size and their shape. So this is an example of what it would look like. So as these particles are flowing out, the first ones, the small, come out first, then larger and larger. And this is an example from our lab where we took a particular type of prion disease, which is very slow and causes symptoms of drowsiness. And you can see there's lots of particles that come out in the small size, but not as many big ones. If we do the same thing with a different strain, a faster disease where the symptoms are with lots of agitation and very hyper, you can actually see fewer small particles and lots of big particles. So what does this mean? Well, both shape and size of the misfolded protein influence disease. They influence the symptoms that we see. Well, okay, how does that work? Well, in neurology, it's all about location. We're very obsessed about localizing lesions. And that's because your symptom depends on which part of your brain is affected. So for example, if your frontal lobe gets affected, it alters personality. If it's your temporal lobe, it affects memory. If it's the back of your brain, the occipital lobe, you can have visual symptoms. And this isn't just in prion disease. Many of you may know people who have had Alzheimer's disease, who may have even had some of these symptoms. In classic Alzheimer's disease, memory is a prominent symptom. Why? The temporal lobe is affected. But there are different types, different subtypes of Alzheimer's disease, different strains. Some affect the frontal lobe, and it's creatively called the frontal variant of Alzheimer's disease. Some affect the back of the brain, called posterior cortical atrophy. So region is really important. So can we somehow correlate protein size and shape with where it goes in the brain, with that brain target? How can we do that? Well, in my lab, we use something called prion disease in a dish. We're all about creative naming. So we actually take pieces of brain, put them in a dish, infect them with prions of different strain, different shape, different size, and look to see which parts of the brain are targeted. And I hope you can appreciate that in each of these pictures, same piece of brain, area of brain, 3 different strains. Everything that's lighting up in green, that's the prions, that's the bad stuff, the big aggregates that build up in the brain. And it depends which strain you have targets a different brain region. Something else we're asking though is, okay, if the prion goes there, does it kill the brain in that region? And it turns out that no, not necessarily. And we're finding that the areas where the large prions build up is not always the area where the brain is dying. Meaning it is the smaller particles that are sometimes more toxic to the brain. So if we put this together, what are we thinking this means? Well, shape may actually influence which part of the brain is targeted, where it goes. But the size may be what's really influencing whether that brain area dies and causes symptoms. And the smaller bits may be a bit more toxic. So this raises the question, could this lead to strain-specific therapy? Maybe if we know something about shape and something about size, where it goes in the brain, what symptom the patient comes to me with, I know, this is the shape that's affecting you. This is the type of treatment we have to target. Well, in my last couple of minutes, I want to introduce another idea of what can we do about this? How can we treat things? And introduce you to a cell in the brain that is called a microglia. And these are microglia. This is what they look like in the brain. They're all these purple cells. They're not really purple. They're just colored that for the picture. But these cells are probing the brain all the time, looking for bad stuff that they can get rid of. They're like the housekeepers, getting rid of things that shouldn't be there. We can actually see these cells in our brain slice culture. So here's one here, sitting there waiting for something bad to happen. When we infect these slices, they change their shape and they become sort of amoeboid, and they start eating things, including prions. So these green spots, these are the prions, and the microglia start eating them. So can we use this to our advantage somehow, if these microglia might eat prions, eat the different proteins? And I invite you to actually check out my student project, so that each of them have posters. I think I saw them back there hiding somewhere. Don't be embarrassed. Where we're trying to ask the question, does size and shape of the protein influence how these microglia gobble them up or not? And can we learn from that and then harness the power of microglia to maybe help with these diseases? So, without going into too much more science detail, I really have to pause and thank those who actually do the work. I can stand up here and show the pretty pictures, but it's really the hard work of my lab members who have persevered with me through, whoa, was that a glitch? Did I forget someone? To really try to make this work go through, because it is difficult and frustrating. Machines break at times. If you're interested, please check out my website. As I said, I do have a TEDx talk where you can see me throw paper airplanes. Doesn't make sense now, but if you look at it, it'll make sense. I want to thank the funding agencies who have supported the work over the years, especially patients, family donors, especially the patients who actually will donate brains to be able to do this work, because we could not do it without them. And just because I like to do these things, I want to leave you with a little philosophical idea of what it's like to be a researcher and to go on this research journey, because it kind of can look like a bit of a jungle, and it'd be hard to find your way forward. And to find solutions, we first have to ask the questions, the right questions. What is the question? And then we have to be creative, and we have to persevere to gain knowledge. And then with that knowledge, we hope we can inspire others to follow in our footsteps. Thank you.
Thanks very much, Valerie. As I mentioned, we're going to have a bit of a Q&A at the end, and so you'll have a chance to ask Val and George and Holger some questions. Dr. Sim showed you a picture of the amyloid protein, and probably members of the society are quite interested in this recent New England Journal of Medicine paper that just came out with an antibody directed against that protein, being one of the first examples of actually improving some scores of cognition in Alzheimer's disease. So that's the same protein that's being targeted. The treatment had a lot of side effects, and I'm not sure it's going to go anywhere yet, but it's the first time we've seen something. And so it just highlights the importance of the specific proteins.
Big stuff.
Yes. Here's a little commercial from the NMHI about Connections. Connections is a collaborative project between the Neuroscience Mental Health Institute and the Faculty of Arts at the University of Alberta that uses images to bring life to relationships between art, expression, neurosciences, and connections. And there's an online gallery. We may be able to have a Connections exhibit at the Royal Alberta Museum coming up. Dr. Scipione has been the steering person that's made this happen in the Faculty of Medicine. And so a little bit more about Connections coming through. Okay, I would next like to ask Dr. Holger Wille to present, and I really appreciate him filling in at short notice tonight. He has a really important and exciting story. He is a professor in the Department of Biochemistry, Faculty of Medicine and Dentistry, Neuroscience and Mental Health Institute member, and director of the Center for Prions and Alzheimer Disorders. And as I said before, you might have heard him on CTV News and in the paper recently, just trying to make the case for the funding of the Prion Center, which we're a little worried about. But he studied biology at University of Hamburg in Germany, conducted research for his master's and PhD thesis in the Max Planck Unit for Structural and Molecular Biology. 2012, he joined the Department of Biochemistry here at U of A, and we're delighted to have him join the group. His laboratory focuses on translating structural biology, so the things that Val talked about, into the design of structure-based vaccines targeting Alzheimer's, Parkinson's, and prion disorders. So a pretty interesting, very novel concept. This presentation, he'll be talking about the laboratory's work to use structural biology to design preventative vaccines targeting these degenerative disorders. So Holger, thanks again for presenting.
All right, thank you for having me. So I'm going to explain to you why we are thinking of making vaccines to prevent Alzheimer's disease, the prion diseases, and the other diseases that come in this domain, and what this means translating structural biology and what are those funny looking things on this screen. Now, we are working on a whole range of diseases, obviously Alzheimer's disease with the two proteins that Dr. Sim already mentioned, the amyloid beta peptide and the tau protein. We also have a project for Parkinson's disease and the protein alpha-synuclein that she also mentioned that's involved there. And all these prion diseases and the prion protein that's causing these diseases. And we're just getting started on a project for amyotrophic lateral sclerosis, or ALS, for another protein in this domain. We got started on this protein with a prion protein. So this started as a prion vaccine project. And because of that reason, we have the most progress on that side. So I'm showing you most of these things on the prion protein. And just toward the end, I mentioned what we're going to do on this end was a vaccine for Alzheimer's disease. Now, Valerie has explained the protein misfolding things, so from the normal shape of the protein and the disease-causing shape of the protein, and that is basically the starting point for our idea how to make a prion vaccine. So we really had to come up with a novel way to make vaccines, because we could not use an mRNA vaccine because there's no mRNA involved in the prion. It's only a protein, so it had to be different. But you cannot use the prion itself as a vaccine, because if you inject it into a person or an animal, you cause the disease because it's infectious by itself. So it causes bad things. So it's a dangerous protein, as you may appreciate from this picture. So this is definitely not suitable as a vaccine. And if we stay in this realm of images like this, this could be a harmless protein, reasonably harmless. Now, how to make this protein into a vaccine for the dangerous one? Well, you would have to give the cow the spots of the leopard to change the surface of the normal protein to make a prion vaccine out of it. And that's exactly what we have done. So this is a very simplified allegory of what we have done on the protein side. And this is where these colorful pictures from earlier come in. Now, let me explain this one. So here on the left-hand side, we have a high magnification image of the normal prion protein. This is the one that we have on all our nerve cells. That's a healthy one. And in the middle, we have a model, which is the idea we had, what the infectious one, the misfolded one, may look like when looked with the same techniques. Now, what we did is we tried to predict which building blocks of this complicated molecules are on the surface of this disease-causing one, and we have highlighted them in green and cyan. And these are 7 building blocks, individual building blocks that come together as we had predicted there. And if we look in the cellular form, the healthy form of the proteins here, you see the green and cyan, they're all scattered all over, they don't come together. So we thought this may be the way to make a vaccine by taking these 7 building blocks that in the disease state come together, as you see in the middle, and put them into a totally different protein that's unrelated and not dangerous that we could use as actually as a carrier for our vaccine. We call this one the scaffold protein, and we use the protein with its good utility that we could easily manipulate in the lab, and this is a protein from a fungus, from a lowly fungus, and the protein itself does not do any harm. We've worked with it for a long time, and you can see on the right-hand side, this is a fungus protein, what it looks like. And in green and in blue are those 7 building blocks that we've taken from the prion protein and put them just into this fungus protein in this precise way. Now, this is a vaccine, how we made it. Now we need to test it and see what it does. Before I do this one, I want to show you this curve, and this is a difficult one for families that are affected by prion diseases, because this is the curve that shows the onset of disease in people who carry a mutation in their prion protein. So this is the onset curve, as we call it, for people who have develop a genetic prion disease. And this, this graph shows the three most common one. For example, if you have the building block 102, and we just numbered them, 102 changed, then you get a prion disease which is called Gerstmann-Streusler-Scheinker disease, or GSS for short. And as you see the green curve, you see an average onset at age 50. Usually people start to have the disease, sometimes earlier and sometimes later. And there's two other diseases, fatal familiar insomnia, FFI, has a curve. Sometimes it starts even earlier. And familiar Creutzfeldt-Jakob disease in purple starts usually a little later. But as you can tell by this curve, if you have this mutation in one of your prion proteins, you usually come down with this disease. So this is the curse of one of these familiar prion diseases. And we know in Alzheimer's disease there's familiar diseases too. We use this information because we know that these mutations give people the disease, and it's nothing we can do about it at this moment. Now we have a mouse as a model that gets GSS. It has the same mutation and the same building block there. And these mice, they start to have the disease, their onset of disease much earlier, of course, at 177 days of age. And they show symptoms like ataxia, circling, they have a hunched back, they start to look scruffy, and they don't live very much longer after the onset of the disease. So this is our model that we used. And if we immunize them, we vaccinate them, but with a fungus protein without those changes, you can see they still get the disease at roughly the same time. So this vaccine there without the changes doesn't do them any good. Now this was a model that we wanted to use. What happens if we give them our vaccine that we manipulated? And this is what happens. So these mice, it's the same mice, they now stay healthy until almost 450 days of age. Looked perfectly healthy. It's the same mouse as the other ones there. They still get the disease in the end, but we extended their health span, as we call it, by 2.5-fold, 250%. So that's a very healthy extension for these mice. Obviously, we don't know if we can use this vaccine and would get the same effect in humans. We know mice are not humans, but something that looks so promising, of course, it is something we are really considering now if this one can be done. And so just as a brief summary for this part, so we have now a vaccine that, at least in this model, works pretty well for this GSS disease. We also have data, it looks equally good for CJD. We also see there in this disease equally good progress, protection of the disease, and extension of health span. So we are now thinking about how could we do clinical trials, and I know one of our big questions is now who would pay for it, because these things are expensive. So we are just at this intersection where we are thinking from where to take it from research in the lab to maybe something that the clinician can handle, because I'm not a clinician. Now, what about a vaccine against Alzheimer's disease? So once we saw the success in the prion vaccine approach, we thought, given how much the similarity there is in the mechanism of this protein misfolding, this thing ought to work with Alzheimer's disease as well. And I'm only having one data slide after this one to explain this one. But before I go there, I just want to show you, of course, you may have seen pictures like this one before. On the right-hand side, you have a typical Alzheimer's brain of a person presumably died from Alzheimer's disease with a shrinkage of the brain throughout. And here you have a brain of a person who died of another cause. After all, I cannot call this a healthy brain because the person died, otherwise you wouldn't see it. Maybe cancer, maybe an accident. The other reason why I want to show this one is because in order to do our research, we rely on having brain tissue from human patients available, both from patients who did not die of a brain disease, so where the brain is unaffected from a disease, and from Alzheimer's disease patients. And I show you in the next slide why this one was so important for us. And the slide is rather complicated looking there, but let me take you through step by step. So up here in the left-hand corner, this gray thing that you see there is again another view of this fungus protein, because we used the same protein that we used already for our prion vaccine, but we made different changes to it. So the changes we made now are the yellow bits that you see in there. So there's building blocks that come from the amyloid beta protein, and that on the surface of the amyloid beta protein. Remember the leopard spots? We did the same thing just for the Alzheimer's protein. We did the same thing for the tau protein. We made several of these. I'm just showing you this very one here. So what we made, we engineered our vaccine to look a little bit like the amyloid beta protein in the brain of patients from Alzheimer's disease. Now we have to test it, of course. And what we did is we took mice, and these were regular mice. This is not a disease model, and we immunized them, we vaccinated them with that. And then we asked ourselves, so these mice develop antibodies, like we developed antibodies against the COVID virus. These mice developed antibodies against this engineered vaccine that we had given them, and now we have to test them, is it any good? And this is where the human brain samples came in. We did an assay where we tested the antiserum, as we call it, from these mice, the antibodies, against brain samples from patients that had died of other causes where the brain was relatively healthy. And this curve that looks a little bit upside down to some degree. This is the blue bars. This is what it looks like when the brain was relatively healthy, no Alzheimer's disease involved in there, and we did not expect the reaction to happen. Now the brown and the beige bars are the ones from samples from patients that died of Alzheimer's disease. Two different forms, a fast Alzheimer's disease and a slower form of Alzheimer's disease. The clinician can tell them apart. And as you can tell, the bars are much lower. And that means the antibodies that these mice made can now recognize the disease material in these brain samples. And we really use just crude brain samples in our assays for this one. And so it can recognize it. And then we thought, what about familiar cases of Alzheimer's disease? And we got some brain samples from these patients as well, from a brain bank. And this is shown on the very right-hand side and the yellow one. And you see they are also recognized. And this is the stage we are in right now. So this is what we have. We have what we call a vaccine candidate that at this stage looks very promising. And obviously we now have to test it some more to see if it can protect. Again, we will start with mice, and we're just getting ready to do these experiments now. If we can see extension of health span or prevention of disease onset in a mouse model. So it looks like our idea of making a vaccine based on what we learned from the prion side seems to be looking very promising for Alzheimer's as well. We have to, the efficacy, as we tell it, does it do anything good in this mouse model? Don't know yet. This one may take a year, this may take 2 years before we have an answer at the earliest. So this is still work in our future. And at this end, of course, I also have to thank the people who do the work. I'm just the boss sitting in the office, but the names here, these are the people who actually do the work in the lab. And on the right-hand side, you see the agencies that give us the money to do this kind of work. And of course, I should not leave here without thanking again the brain bank and the patients and the families who agreed to donate the brain tissues, because without this one, we could not do this research. And I thank you for your attention.
Well, a big thanks to our speakers. We're going to spend a little bit of time now with Q&A. If you have some questions you'd like to ask the speakers, and we have maybe 10 or 15 minutes we can use for this. And I think we have a moving mic, right? Yeah. So anyone? Don't be shy, and there's a...
Yeah, I was just wondering if you can, within the same aspect that you were talking about, can you enhance the density? Sorry. Thanks. Yeah, I was just wondering if you can enhance the density and the activity effectiveness of the microglia with respect to the the topics that you were talking about.
Yeah.
So the question, can we increase the density or the concentration of microglia, essentially?
Do you affect them in any positive manner to
Right.
accelerate your design?
Excellent question. So many people have been studying microglia. We're new to the business, and there are actually many ways you can enhance and stimulate and promote the microglia to, increase activity and clear things. The trick is it's a double-edged sword, because turning these things up and letting them loose has side effects that we sometimes don't want as well, because they can gobble up. Suppose your neuron is a little bit damaged from the disease, that microglia might actually help clear up your neuron too, and so could help lose neurons. So it's a balancing act, as is often the case in these situations. It's the balance of how active do you want the microglia, and also timing. When do you want to clear this material? Because something we didn't touch on is in all these diseases, the protein misfolding starts years before you see any symptoms. And it has to really build up in the brain, and only then do we finally see some symptoms. And if there's a time to get rid of the protein building up, it's probably way back before we have symptoms, but then we don't know. So that's one of the challenges. But it's an excellent question, and I think if we can be more precise in which parts of the microglial action we can enhance and not do it at the wrong time, there might be some hope.
I have a few questions in terms of trying to understand exactly the mechanism behind this. So it might be a little multi-part. But when I was looking at your images up there, it appeared to me that what happens is these misfolded proteins, the prions, tend to clump together. And so if I'm correct, then the use of the vaccine is basically to act like a sponge on these misfolded proteins. They attract each other to that the scavenger protein from the fungus that you've instilled in the patient. Is that an accurate way of understanding it?
Yeah, we still have quite a few questions about what exactly is the mechanism behind it. Our initial idea was that, for example, the antibodies that the vaccine elicits in the immunized animals, and presumably later on in humans as well, if we ever get to this level, would be, for example, preventing this propagation happening, the seeding that Dr. Sim had mentioned. We have some preliminary data, and we have to work some more on it, that tell us maybe it's not the antibodies after all, but maybe it's the immune cells, which is the second big component of our immune system. And they are, of course, very active as well and contribute a lot to our immune response. And now we are looking at it, maybe it's the immune cells, so maybe it's a killing function of this one. Obviously, again, there's always a question, what are they killing? At what stage does this happen? And we do not have any answers to that yet.
All right, we're going to go to some online questions here from our streaming audience. First question here is from Laura. Does prion size and shape correlate with the age or onset of heritability?
Thanks. I always teach my students to answer questions like that. That's a fabulous question, which means we don't really know. There are some genetic forms of the disease, like fatal familial insomnia, which is a terrible sounding name for a disease, where the degree of aggregation, the size and the sensitivity of the aggregates is different and actually less than diseases like, I'm going to try to say Gerstmann-Straussler-Scheinker as a non-German speaker, where another genetic disease where the size of the aggregates is huge and forms these massive clumps in the brain, but without really clearly changing when the disease starts. Another big challenge in genetic prion disease, there is a huge range of onset. I think Holger showed a slide of the range of onsets, and I have met family members where in their family someone has died of the genetic disease at age 40 and a sibling at age 80. So it makes it very much a challenge to understand why is one faster than the other. And I think it's probably oversimplistic to think it's only the protein, because as people, we all have our own variability. Why does someone get COVID and someone not? Why do you get a sick cold and I get less of a cold? There is too much variation within us that is also influencing it, maybe through the microglia, maybe through ways that we are clearing the proteins, because once the misfolding starts, it's a race. And if the body can just keep clearing it, perhaps you can last a bit longer. But if you're not as good at clearing things, then you lose out. And that relates a little bit to what we tell people who have Alzheimer's disease, or how to try to slow down or prevent Alzheimer's disease. We try to promote what we call cognitive reserve, use it or lose it philosophy. And we say more social activity, more cognitive activity. And it doesn't just have to be, Sudoku or whatever, but use your brain. And social activity, cognitive activity, and physical activity, trying to keep your brain as healthy as possible. That is clearly influencing its overall health and ability to clear the protein aggregates over and above what shape and size they are.
I think we've got a couple more online questions at least, but I wanted to bring George into this just a little bit and change the direction just slightly. You saw from Val's slides and Holger's slides the students and the people that work in the lab and get this done. And so, people think, well, where does funding go for research? What bind alleys does it go down? And 70 to 80% or more is to fund people to learn about this and work on it. And George, I think the society views things in a similar way, but I wondered if you wanted to comment on where you saw the society and the support of trainees to solve Alzheimer's disease.
Thank you. Well, we approached funding of research in a variety of different ways. Through our national office, which we've been working with for close to 2 decades now, we fund early researchers earlier in their career, help them get some seed funding for those projects we see outside, for example. I'd encourage you to go and look at those boards, because those are the future for our research, our big research. And when those people are in their prime is when we're expecting this big wave of new emergent, activation on this disease. So that's one stream. The other stream is actually working with donors that have some suggestions on where they want the areas they want their funds to go to. So we look at different outcomes, whether it's quality of life or clinical work. We funded a few projects through the Prions Institute previously that was really focused more on the, I would say, the preliminary, front end research, which is crucial. And now we're doing a little bit more work on a peer-reviewed project, which, as I mentioned earlier, $5 million from the foundation, our society, and plus another $5 million from other donors. And that's really looking at quality of life and really different modes of research. So it's a mixture, because it's not just one thing. And we're constantly engaging with all our stakeholders. And by stakeholders, I mean people living with dementia and their families, as well as researchers and government, to understand what's important and what can we do and how do we have a balance. So much like understanding the disease is a bit of a balance, and sometimes the treatment has some negative side effects. We're also looking at balances around the here and the now versus the longer term and quality of life. And much of our work is really focusing on helping people in the here and now. So the bulk of our staff are engaged in supporting people who have their own individual journey, for lack of a better description, and whether it's understanding what resources are available to them, how to work, how to take advantage of systems that are in place already, and also how to clear information from other jurisdictions as well and share that information. So I'm hoping I hit that point. And is there any other questions for me or our colleagues?
I have a question.
Okay, you need a microphone. I'll repeat your question.
So they're looking at prevention and a cure. How would the Alzheimer Society adapt to the emergence of a cure and prevention?
So the question was, as researchers are looking at cure and prevention, how do we adapt to support that? Well, I mean, I would love to have a day when I'm working for the Alzheimer Society and we close the door because the disease has been completely treated and we don't have to worry about that. But the reality is that that's maybe a generation or two away. There are things on the horizon that give us hope, and our tagline is hope for today, help for today and hope for tomorrow. So in that regard, we're trying to work with people who are living in the today, and the reality is it'll be probably their children or their grandchildren that are going to benefit from the research that's undertaking today. But we won't have answers or cures or viable treatments until we start doing the work today. And I want to emphasize that we do know that if you take some preventative lifestyle interventions, you can delay the onset of many symptoms. And so if we were looking at quality of life, which is a large part of our conversation with clients, if you can delay the onset of symptoms a decade, well, that makes a big difference. And so there are techniques and strategies that you can employ, and I encourage you to come to our website or come to our focus groups or to our support groups and learn more about that. Because it's one thing to fund research and put it in the hands of very competent people. It's another thing to take control of your own life and see what you can do to help mitigate some of those challenges that are coming. Okay.
We're going to do a couple more online questions here, and then we'll flip back to the room. We'll alternate back and forth here. The next question that I have here, let me just have a look. Do these theories apply to nerve and brain injuries, such as concussions?
So I'm not going to touch the nerve injury. You can do that. That's an excellent question. I'm not saying that because I don't know the answer this time. And in concussion, actually, it sort of made popular, if you will, in the media with the cases of football players who've had concussions and had a lot of psychiatric issues and suicides following. We know that in some people, repeated trauma can trigger misfolding of a protein. Which protein? Tau. One of the two proteins that we see in Alzheimer's disease. Tau occurs in actually a number of different diseases as sort of an extra protein misfolding that actually may be more toxic to the brain. So concussion, at least, can certainly lead to this buildup of protein misfolding and that type of trauma. Nerve injury, I don't know if we can say in the same degree. On an isolated nerve, we see it much more, the spreading, it can progress through nerves and to the brain, but the symptoms that develop, we see in the brain. Hopefully that covered it.
Wonderful, thank you. One more question online, then we'll flip back to the room and continue alternating here. We have one question related to the development of the COVID vaccine. What would it take to move forward a vaccine as aggressively with the COVID vaccine as it would for the Alzheimer's vaccine? Would it take money?
Yeah, so contrary to popular belief, the development of COVID vaccine did not take 1 year. It also did not take 2 years. It took actually closer to 20 years because the technique to develop these mRNA particles, the lipid particles, to put all these things together, that's research for about 20 years. What happened when SARS-CoV-2 showed up is that they took this sequence, they sequenced this virus, exactly what are the building blocks in there, and plugged it into the existing idea of how to make an mRNA vaccine. And it worked. Obviously, they had to test it, so the testing was very rapidly, but to say that the COVID vaccine was developed in a year or two is not true. So with the Alzheimer vaccine, we are basically in year 4 or 5 now, so we had to do some development first to get it to the stage where we are. So if you say it takes us another 10, 15 years, that sounds about right because we are now going to test it in mice first. So this will take a few years before we have this one. How much longer it will take before anyone would let us to go close to humans with this one, I don't know, but it would be a long time. So That's why when everyone thinks that COVID vaccine was developed so rapidly, that's not true. This was just this final step of putting the right sequence in there to make it specific for COVID, because otherwise they have mRNA vaccines for some other viral diseases that most of you will not heard that there is a vaccine available. For Ebola, there's an mRNA vaccine that fortunately we don't have to worry about. The disease is not present here at the same level, but the vaccine exists, and then this knowledge was used to make the COVID vaccine.
I just think this is a question for George. Are the support groups and focus groups back in person?
So the quick answer is yes. But one of the benefits of COVID is that we learned how to use technology a little bit differently. So we have quite a few people who still have some reservations around coming together in support groups, so we still do online support groups. And we're seeing a request for hybrid support groups. So if there's family members in different jurisdictions, we can have face-to-face with our facilitators on site, and then we still have the capacity to bring people in. So yes and more was the answer.
Don't sit down, George. Not to be alarmist, but you mentioned that you expect the incidence of this to grow by 300% over the next period of time. I'm sure we all appreciate that our healthcare system is undergoing a few stresses right now. What would that look like, potentially? Because my in-laws are going through this wonderful disease, and I see what it does to them. And I just have concerns about our society being able to support people who are going through this. If we're looking at a 300% increase, so that means going from like incidence of 100 per to 400 per. Is that, have I got the right math on that? Yeah. So not to be alarmist, but what could this look like?
So be alarmist. I mean, the reality is there's a couple of different things at play. First of all, there's people smarter than I that are going through this modeling. But we know that the more people are aware of dementia and it's being diagnosed more frequently. People are living longer. And so as a natural consequence of living longer, you're more likely to end up developing some form of dementia because that's the largest correlation. But as our populations grow and age, that's also another natural occurrence. I think there's a variety of different things that are going to come to play, whether it's environmental or whether it's just because we're paying attention. It's going to be there. But the big point that you're making is that, I think you're making is, how can our healthcare systems across Canada and around the globe hope to engage and support people and their families? And the real answer to that is, if you do the same thing over and over again and you expect different results, I think there's a room for you in a different building. And so this is a wake-up call to find out what's going on. So I'll give you a little bit of a snapshot. 2 weeks ago, my mother passed away from consequences of Alzheimer's disease. And like a lot of people in her circumstance, there was lots of falling, a lot of confusion, other things. And in the last 12 months, she probably spent 10 months in hospital. An acute care bed, the absolute worst place and most expensive place for a person with dementia. We need to come up with a better system. And so part of our research and part of our focus as a society is to bring attention to the issues and to look at policy change and how we can make things happen and things like that. And not to do the same thing over, and not just to push people back in their homes. Now, keeping people in communities, I think, is a good idea. Supporting their families and the communities to help these people is the best idea. And so we have to look at that, and there is really policy decisions and discussions that have to happen. And it's only going to happen when people make enough noise to their elected officials to say, this is important to me, and I'm a voter, and my mother may have dementia, but there's 18 people in my immediate family, and we're all voters. And so we need to have that kind of dialogue. And I'm cringing a little bit because I've got a couple of board members in the audience. And so, but the reality is we have to have this dialogue. I think the other thing that's happening is that there's been some great work talking about mental health in Canada and around the world. And so many people a decade ago, if they had dementia, there was a stigma associated with that. And, you know, and I think as those stigma points move along, then we'll become more and more aware of things. When my mother was first diagnosed with dementia, she did not want me to tell my siblings or her siblings. Well, that's just her generation to understand those things. So all of those things are going to come together, and we need societal change, and we need to talk about it. I hope that answers the question.
All right, one online question here. Have researchers been able to identify if there are different misfolded proteins in other forms of dementia, for example, frontal temporal dementia?
Yes. But it's not one protein. And guess what? There are different subtypes of frontal temporal dementia and different proteins that build up in it. One of them is, guess what? Tau. Another one is TDP43. Another one is FUS. So they come up with interesting names. And even if it's tau, there's like 6 different versions of tau that can fold into different shapes. So it becomes quite complicated. And one more kicker, sometimes you have a combination of pathologies. So it isn't just one protein. When you actually look, you can see more. And I think this highlights another really important aspect of understanding the disease. We only know so much and can predict so much with testing, imaging, blood samples, spinal fluid tests. At the end, the only way to really know exactly what the pathology, what proteins are involved, we need to look at the brain itself afterwards. And that's something that's a very precious resource, but it's really necessary for us to understand from people who do pass on from one of these syndromes to really understand and answer that question. But absolutely, protein misfolding is driving those frontal dementias too. Thank you.
On several moments, there has been a comment about the need for brains. I suspect many of us here have loved ones who have Alzheimer's or another form of dementia. I have absolutely no idea how I would ensure that my husband's brain can help further research. So what does one have to do in order to make brains available?
This is also an excellent question. And what we really need are what we call brain banks, which is sort of a crude way of saying a place where we can store a bunch of brains from people who have donated. And that actually requires resources and money to have the freezers and the wherewithal to do that. There is some storage happening at the U of A. There is a brain bank in Calgary. And it really, though, has come mostly from universities that have been willing or able to do that. And we have talked in neurology a bit about trying to, can we support or find a way to actually organize and have such a bank? So it's a very valid question because there is a research need, but the pathway to donation such that it will get somewhere, I don't think is very well ascertained. If a patient goes to autopsy, the best one can do, there is a consent within that form that says, do you consent to have materials for research purposes? And that's really the key. And then it opens it up to a researcher who has a question, who has a specific need for that particular brain. And that's, I think, at your level, where it has to start, that yes, we want the brain to be available for research purposes, for autopsy. Then you have an answer of exactly what was the dementia at the heart of everything. and it can be made available. Will it 100% go? That is always difficult to say because it depends on the researcher's specific question. And I don't know if you have more to add on that, but that's, for me, where I have been getting samples as I need them, has largely been from the Brain Bank in Calgary, through pathologists, colleagues here in Edmonton, and even for the prion-infected brains, there is a central brain bank actually in Ottawa. It's housed in Winnipeg, centered in Winnipeg, housed in Ottawa, where it's a bit more organized. But yeah, it's a very good question, and it's not as well oiled a machine as it could be. Do you have a comment on that?
Yeah, there is an effort to collect human tissue here that Dr. Ritchie, a hematologist, has set up. And I mean, it's really important where these are, to have them supervised, to have them protected, to have alarm systems on the freezers. And it's at risk, just like the Prion Center itself. It's at risk with the funding cuts that are going on to support this. It takes resources. So, yeah, I think tonight is a lot about the partnership of the society and NMHI. I think there's room to help, if you're so inclined, to support things at a variety of levels. And I'm glad George talked about that as well. NMHI lives a lot on voluntary work. We could really use the help. Holger's been talking about the troubles with the Prion Center, keeping it alive. To be able to do that kind of work under isolation conditions, to manage those proteins, it's very tricky. And the Prion Center is in a little bit of trouble right now. But hopefully we'll get through that. I also want to reiterate what George said and the question about how our healthcare system is going to sustain things, and Val may want to comment too. I mean, we hear about the wait lists going into the emergency room for anything that you have to wait for. But when Val and I are on the neurology service and we're spending time in the emergency room, the disproportionate number of persons dealing with dementia that are in emergency rooms at any given time, they've fallen, they've broken their hips, they have urine infection, pneumonia, it's massive, and with a 300% increase. I think that's a major issue. Sure, another question, yeah.
Opportunity. My question was about brain donation. My mother has Alzheimer's, she's end stage, and we're trying to have that organized. We believe she passionately would want to pursue this. The challenges we've been having have been incredible. So we want to. Doctors don't know, the long-term care facility don't know, like, it's impossible. There is some good information on, George, on your site that refers about needing to donate it to the Maritime Brain Bank. So I'm interested in these other options that have been explored today, but I'll gladly share my journey, but that would be one barrier that I would recommend. I think there's probably a lot of people that might be interested. Recognize that it's an extremely personal decision, but it's been two of us working, calling multiple people, and we've actually figured it out and educated the long-term care facility so that they know, but it's not easy.
Thank you for that comment. I think we'll wrap up the questions now. I think Holly wants to present something here.
Certainly just a quick presentation to our guest speakers tonight, and just wanted to say thank you so much for spending your time with us and educating us on developments in Alzheimer and dementia research. It's been so exciting to hear about what's at the forefront, and so pleased that we could be here at the University of Alberta in front of a live studio audience, if I can say that. It's nice to be in person, and also our online audience, thanks so much for joining us. But I do have just a small thank you gift and gratitude for spending time with us tonight. So thank you.
George, I think yours is outside. Sorry. All right, let's wrap up. There's an opportunity to come outside and see some posters and socialize. It's good for your synapses and brain to do that. I'm going to finish with a slide from Connections, as it turns out, our project on neuroscience and art. And this is one of the images in the Connections book that's come out by Hunjung Lee. And I think she's in the audience right here. Yes. I'll just quickly read it out. This piece represents age and time disorientation, which is commonly experienced due to Alzheimer's disease or other types of dementia. Challenging the dominant narrative of decline, this image urges us to rethink living with dementia in different ways of being and living. And it's a striking image. Thank you. Well, again, I want to thank everyone for great questions and for staying and paying attention. And I think it's been a terrific event. Again, a huge thanks to the Society for coming up with the idea, and for Ramona for helping to organize it, and other members and our speakers. And with that, I think we can call it a night. Thank you.